ecology. Sampling methods. Okay, sampling methods in a population ecology. To know the number and distribution pattern of plants or other organisms in a desired area, it is not possible to count all of them by manually. Then counting of few representative part of the study area is called as sampling. The sampling method is nothing but density is measured by this method, and it depends on a type of organism and its a natural abundance and the distribution. Now the next slide. So biologists need to collect data or a data in a field. This data needs to be collected in a standardized way. So two methods of doing these are a transect and the quadrant method. Now the use of quadrant. So quadrant are used for sampling purpose. They are squares of a set size placed in a particular habitat such as a rocky shores or a forest floor. The plant or an animal within a quadrant are identified and their number recorded. The photos of individual quadrants along with the species information often form a baseline monitoring data or are used to measure changes in a species and habitat over time. The quadrants are square frames of wire, usually 0.25 meters square. These are placed on a ground to look at the plants or slow moving animals within them. When looking at a plant in a quadrant, the following sampling can be used. So the number of individuals in a species, species richness and percentage cover. As we know that the quadrants, so the quadrant method, this is the sampling method, is a classic tool as a series of squares or quadrant of a set size mainly used in ecology and geography to study a plant population or for the objects of a large area. A quadrant is a suitable frame, mostly squares, for a sampling of plant of varying size areas. The sampling of plant or slowly moving animal like a snail can be done using a sampling square called as a quadrant. The plant ecologist R. Pound and E. F. Clements developed the use of quadrant method in 1900 to study the plant population. This method is used to find out similarities or differences in composition and structure between various plant communities. So the quadrant, it is of four types: list quadrant, list code quadrant, chart quadrant and clip quadrant. In a list quadrant, the list of species present in a particular quadrant. List code quadrant, it includes the population of each species in every representative quadrant. Chart quadrant, it is a graphical presentation of area covered by a grass, mat or mosses, etc. It helps to show a change in community in the future. Clip quadrant, a biomass or a weight of each species is studied in this method. Fresh or a dry weight of uprooted individual or branches or fruits were recorded. Grazing animals shows the effect on vegetation, so it should be studied by fencing the quadrant by proper material. Now, the quadrant enable a comparison between the different areas of survey site. A quadrant is a frame area. The area within quadrant is a section into set of number square or the intercept. Now, the using quadrants, how to use? What you have to do is mark out an area to be sampled. Place quadrant, it is 1 meter square or 10 meter square randomly within the area. Count how many individuals are inside each of the quadrant. Calculate the mean number of individuals per quadrant. Proper size or the population size is equal to mean into a total area of each quadrant. Frame quadrant, a square frame divided into grid of 100 small squares. Place on area or investigation after random assignment of coordinates. A number of individual species is recorded in each quadrant. Can calculate the percentage cover of the plant species. How much of quadrant is covered in a plant species quicker as don't count the individual plants. Small area is included, that is 1 meter into 1 meter. Larger area can be marked with a tape measure. Now, how many weeds are present in this field? So total area of field is 200 meter into 15, that is 10,000 meter square. Area sample, that is 10 quadrant into 0.25 meter square, that is 2.5 meter square. So number of weed is sampled in area is 56. Area of quadrant, that is 0.5 into 0.5 is equal to 0.25 meter square. Now here you can see in this picture, so plant species, yellow dandelion, pink flower and the grass, small, medium and the large quadrant. So in a small quadrant, the yellow dandelion is not present, so 0 is there. After that, medium quadrant 1 is there and large quadrant 4 is there. In a pink flower, so a small quadrant shows the 1 pink flower, medium quadrant it shows the 3 pink flower and large quadrant it shows the 8 pink flower. Now the grass, so a small quadrant it shows the 4 number, medium quadrant it shows 10 and the large quadrant shows the 25 grasses. Now this is second method, that is transect method. So line transect method and belt transect method are two types. In this line transect method or intercept sampling method, systematic sampling is used when you choose where to take samples because you are investigating a specific pattern in an ecosystem. The most common kind of systematic sample is a transect, where the samples are taken along the straight line to see what changes there are along the line. The transect usually follow an environmental gradient, such as down a rocky shore, into a forest or down a mountainside. The transect should be a few meters long as a few hundred kilometer or 100 kilometer long. A line transect, the organism touching a piece of string stretch along the transect are recorded. The belt transect, quadrants are placed in an interval along the intersect and the organism of each quadrant are recorded. So line transect is a quick but can be unrepresentative, while the belt transect involve more work but can generate a more complete data. Now this is the diagram of belt transect. As we know that using sampling using the belt transect, okay, so here a better method of line transect, a belt transect is made by laying out two parallel strings, perhaps 1 meter or less apart, record the plant between them, or placing quadrant continuously or in a regular interval along a transect line, that is quadrant 1 2 3 4 5 like that. So what we have to do is we require a string or measuring tape required to make out a line. This method records the information of abundance of particular species as well as a presence or absence of it. In this transect line come out across the survey area, and a quadrant is beginning mark point on a line. The individual, it may be a plant and animal, it is identified inside the quadrant and their abundance is recorded. The animal can be counted or collected to find the percentage of plant species. Cover is the area of quadrant occupied by the above ground part of the species when viewed from the above. Like now the sampling by using the line transect. You can see here the line transect, a string or measuring tape to mark out a line, commonly used to measure a percentage of vegetation coverage, woody debris, mat, plant or animal abundance, etc. Now, a line transect is useful for examining the effect of zonation or the biodiversity. The graph shows the presence of each species along the line transect. In this case, the line transect crosses a stream. Now, distance along the transect measured in a meter. A line transect does not reflect the density of each species along the line. It is only useful for basic analytical tool. You can see this is the line transect. So percentage gross is equal to 1 plus 1.7 plus 0.6 plus 0.8 and 10 units total. Okay, you can see here. So this is the 10 units total. So we took the 10 units in total. So what we have to do, we have to add all these points for the addition. So 1.0, after that 1.7, after that 0.6 and 0.8 in a red only. So 4.1 divided by 10, okay, and into 100 it gives 41%. Now for grass, so what we have to do is 0.9 plus 1.3, right? 0.9 plus 1.3 divided by 10, and here is your answer. So what you have to do is you have to solve this in your book. So this is about the belt transect method and the line transect method. Thank you.